Hi all, Tony here from Immortality Cryptocurrency. Today we want to discuss a long-held strategy and pathway to cure aging. Saccharomyces cerevisiae. Yeast. Yeast studies. Why yeast single cells age and die. Scientists claim that they have already identified the root causes of aging. Extensive studies in yeast cells place the causes of aging in two places. One, a gradual decline in the stability of the nucleolus, more specifically the overproduction of ribosomes, or extrachromosomal ribosomal DNA circles, ERCs. Ribosomes are the basic building blocks of proteins, while the other half age due to dysfunction of mitochondria, the energy production unit of cells. There have been many genes identified with longevity and aging. One such database is the longevity map at senescence.info. But it can be suggested that one cure is the activation of stress-resistant genes. Paper titled Life Expectancy of Yeast Extended to 800 in Yeast Years, No Apparent Side Effects, identifies two genes, Ras2 and SCH9. Ras2 is known to downregulate stress responses, demonstrating that for longevity, the ability to recover from stress is at least as important as the ability to mount a stress response. While SCH9 effects may be explained by constitutively active oxidative stress response system preventing accumulation of age-related mutations, perturbed sphingolipid levels, or, more likely, a combined effect of these factors. In mainstream culture, many therapies such as caloric restrictions and cryotherapy attempt to activate these stress response genes. Their efficacy and effectiveness is debatable. Another paper, A Programmable Fate Decision Landscape Underlies Single-Cell Aging in Yeast, suggests two modes. Half of cells produce daughters with an elongated morphology during the later stages of lifespan. In contrast, the other half continuously produce small, round daughter cells until death. Thus, aging mode 1, nucleolar decline, as described, and aging mode 2, mitochondrial decline, as discussed previously. The same mode has been expressed several times by several different groups. Age-dependent nucleolar enlargement is related to instability of ribosomal DNA, the conserved lysine, the acetylase SIR2 encoded by a well-studied longevity gene, maintains ribosomal DNA stability by mediating ribosomal DNA silencing, while a decrease in heme abundance and HAP activity may drive mode 2 aging. In another paper, New Technique Reveals Causes of Aging in Yeast states, our idea is that the increased production of ribosomal proteins is good for young cells, says Veenhoff. It allows them to grow and mature quickly. But this growth program somehow keeps running and eventually becomes a negative factor. The paper suggests the overproduction of ribosomes elongates the cell, causing cell death. In a protein that extends life of yeast cells, the researchers discovered that a gene called NDT80, for example, is activated at the same time that rejuvenation occurs. When they turned on this gene in aged cells, they were not reproducing. The cells lived twice as long as normal. In aged cells with activated NDT80, the nucleolar damage was the only age-related change that disappeared. That suggests that nucleolar changes are the primary force behind the aging process. These papers do not read like theory, so the science is getting stronger for yeast cells at least, determining if the same is applicable in human beings and identifying precise ways of activation is essential to finding out. The technology to manipulate gene expression of one or more genes appears seriously lacking though, and one highly developed gene regulation system could potentially be portable across many conditions, including aging. This would be preferable as there are too many genes and too many pathways. Here is a short video describing the situation.
This is a lecture on the genetic regulation of longevity and what we know about the genetics of aging from studies in the model organism Cerevisiae or yeast. And this lecture is for September 4th, 2020. And a yeast cell will generate about 20 to 30 daughter cells, or those identical budded cells, in its lifespan. And so the more bud scars, or the more times a yeast has replicated, the older that yeast cell is. And so using the number of bud scars, which you can see here in this EM, can actually help you identify how old the yeast is, at least in terms of replicative age, or how many times it's reproduced before. And although bud scars are used to kind of correlate to age, there's no evidence that the formation of a bud scar actually causes anything to do with aging. It doesn't cause this rapid decline that you can see here, right? So it's correlative with age of yeast, but it's not causative. It doesn't actually age them. And what's interesting is that the genetic mechanisms that do control replicative senescence, or this basically decrease in the rate of reproduction seen around 20 bud scars, is still pretty much unknown. And so we don't really know what genetics control the senescence in yeast cells as of yet. And we know that bud scars correlate with yeast age, but they definitely don't cause senescence. So what do we know? We do know that there are these particular forms of DNA, particularly called ribosomal DNA or rDNA, that are in the nucleolus of yeast cells, and they can form what are known as ERCs, or extrachromosomal rDNA circles, via recombination. And so rDNA is very repetitive, and each one of those repeats can be seen here as basically a thick line. And the rDNA, as I said, is located in the nucleolus, which forms ribosomes. And in a young yeast cell, this DNA is able to replicate relatively easily. However, as yeast cells age, there's a higher chance for recombination events. And those recombination events produce these ERC circles, or these extrachromosomal rDNA circles. And those ERCs can accumulate over time. So an older yeast cell will have a larger accumulation of ERCs than a young yeast cell. And that's because this process of excision, as well as forming an ERC via recombination, the chances increase over time. And the accumulation of ERCs is not only correlated with the age of the yeast cell, but also with replicative senescence, or basically the stopping of replication for a yeast cell and its inability to bud and form new yeast, as well as with a shortening of lifespan. So if you take a young yeast cell and then you take some isolated ERCs from a different cell and put them into the young yeast cell, you can automatically shorten its lifespan. And so the presence of these ERCs is enough to shorten the lifespan of a young yeast cell. And that's likely through fragmentation of the nucleolus from the accumulation of these ERCs, which will ultimately lead to cell death. And so why do ERCs lead to a decrease in lifespan? Why can you take them and then put them into a young yeast cell and see this decrease in longevity? One other protein that's involved in the formation of ERCs and kind of preventing that is what's known as the SIR2 protein. It's encoded by the SIR2 gene, and that SIR2 protein is a histone deacetylase, which is a modifier of chromatin. Right? And so histone deacetylases remove the acetyl groups from histones and turn off gene expression. And so in a young yeast cell that has wild type or normal SIR2 gene expression, you'll see the SIR2 protein, this histone deacetylase, basically deacetylating or stopping expression of rDNA, or that ribosomal DNA. And in doing so, it actually stops the formation of ERCs. And so SIR2 decreases rDNA expression, and in the process decreases formation of ERCs. However, as the yeast age, you can see the removal or that loss of SIR2 expression that then allows the rDNA to become expressed and start to form ERCs, which will ultimately lead to cell death. And so you can imagine that if you have an overexpression of SIR2, or a lot more SIR2, you can extend lifespan by keeping the rDNA expression suppressed for a longer period of time. So that overexpression of SIR2 will increase lifespan by decreasing ERC formation. Whereas if you delete SIR2, you see the opposite, where you're promoting formation of ERCs and therefore decreasing lifespan. And you can actually see that here. So this little delta sign, or this triangle, after SIR2 implies a deletion of the SIR2 gene. And so a deletion of SIR2 will increase ERC formation and therefore decrease lifespan. And so you can add even another level of regulation to this genetic pathway. So first, we linked increasing ERCs to aging and decreased lifespan. Then we linked SIR2 decrease to an ERC increase to decrease lifespan. And now we can add in the environment. And so SIR2 expression, or the expression of the SIR2 gene and the protein as a result, is highly subject to the metabolic environment. And so when there's a lot of food around for the yeast, they have a high rate of metabolism. And that high rate of metabolism means a lot of cellular respiration. And if we think way back to cellular respiration, one of the main byproducts through the Krebs cycle as well as glycolysis is NADH. And NADH is a reduced form of this molecule here called NAD+. And so when there's a high rate of metabolism, all of this NAD is converted into NADH. And that low concentration of NAD inactivates the SIR2 gene expression. And when you don't have SIR2, you don't have the histone deacetylase, your chromatin stays acetylated, our DNA can be expressed and make a lot of ERCs. However, in a food-poor environment or with a little bit less nutrition, the rate of energy metabolism decreases, which means respiration decreases, which means less NAD plus being converted to NADH. And as such, that NAD plus concentration goes up. NAD plus can activate SIR2 expression. And that means you're making the histone deacetylase protein. That histone deacetylase can remove acetyl groups from chromatin. And when it does that, it turns off expression of rDNA. And it decreases the formation of those ERCs. And when you have less ERCs, you can extend lifespan. And so altogether, a food-poor environment can actually extend lifespan in yeast. Which is sort of interesting to think about, because we generally think more food means more growth, which would mean better lifespan. But actually, in the case of yeast, we're talking about amount of replication, right? And so in a food-poor environment, we can kind of slow replication down. And every time you slow down that process of the yeast replicating, you extend the yeast lifespan as a result, because it can only divide so many times, right? And if you prevent it from dividing quite as quickly, it can live longer. And so having a slightly less nutritious environment can actually end up extending the lifespan of the yeast via this pathway and SIR2 and ERC, a decrease in ERCs.
That was Dr. Amy from Biology for All. There are currently no guaranteed ways of ensuring our DNA do not aggregate in the nucleolus, neither are there any guaranteed ways of ensuring mutation-free mitochondria. Two essential technologies are required. For immortality, gene therapy precedes chemical pathways for both rDNA silencing and mitochondrial DNA mutation correction, as well as silencing and expressing key genes. Immortality cryptocurrency, thank you very much.